Hi, my name is Hannah Schmidt, and I'm going to be discussing cases 2 and 3 in this week's Case Connections. So, to start with, in case 2, this is a case of a 4-day-old infant, patient A, who presents with elevated phenylalanine levels of 6.6 milligrams per deciliter. In conditions where phenylalanine is impaired, it results in tyrosine becoming an essential amino acid because normally tyrosine is synthesized from phenylalanine. Tyrosine is a substrate for thyroid hormone synthesis as well as for the production of melanin. The likely cause of PKU in this patient is a defect in the phenylalanine 4 hydroxylase gene known as PAH gene. The PAH gene provides instructions for making phenylalanine hydroxylase, and mutations in this gene can result of the buildup of phenylalanine to toxic levels. The liver of aborted fetuses has been shown that after the prenatal diagnosis of PKU, that there was no detectable phenylalanine hydroxylase enzymatic activity or immunoreactive protein, but both were found in control specimens of the same gestational age. The size and amount of mRNA were normal, and this confirms it is a genetic diagnosis and that mutations affect the translation of the protein. PKU is autosomal recessive, and both copies of the gene in each cell have mutations. The phenylalanine diet restriction was implemented in this patient, and the response was a decrease in the phenylalanine levels. Hyperphenylanemia is caused either by PKU or BH4 deficiencies. BH4 is a cofactor synthesized from GTP. It is required in the metabolism of phenylalanine, tryptophan, and tyrosine. It is oxidized during hydroxylation reactions. PAH converts phenylalanine to tyrosine using BH4 as a cofactor. The pathway with BH4 is responsible for most catabolism of phenylalanine and involves disposal of 75% of dietary phenylalanine. The remainder is used for protein synthesis. BH4 testing historically was performed to distinguish between PAH deficiency and BH4 deficiency. However, the BH4 deficiencies are a class of disorders, and now other labs such as the Paterins, Neopterin, Biopaterin, and Primopaterin are measured, as well as dihydropterin reductase, DHPR, to distinguish between the different types of BH4 deficiency disorders. Now, it is performed still in order to determine if the patient is responsive to BH4 because this can be used as a potential treatment option. So the test in this case was performed on both infant A and infant B. In patient A, the phenylalanine levels rose after administration of BH4. In PKU, there is either complete or partial inability to convert phenylalanine into tyrosine. Since administration of BH4 had no impact on phenylalanine levels, it would indicate a complete inability to convert phenylalanine to tyrosine in a completely non-functioning enzyme. Since no response to BH4 is seen, this would not be a treatment option used for this particular patient. In patient B, the phenylalanine levels rapidly declined after administration, indicating responsiveness to BH4. There are variants of PKU that allow the enzyme to retain some activity, which is known as variant PKU or non-PKU hyperphenylanemia. Deficiencies that lead to decreased synthesis of BH4 can present similarly to PKU, so this infant could also have one of those BH4 deficiencies. There isn't a scale on the graph, but if the phenylalanine levels fall over 30% at any point, it indicates BH4 responsiveness. If there is a rapid fall within 2 to 6 hours, it suggests BH4 synthesis disorder, which would either be primary BH4 deficiency, guanosine triphosphate cyclohydrolase, or 6-pyruvyl tetrahydropeptrin synthase deficiency. If phenylalanine drops to normal in 24 hours, this indicates dihydropyperidine reductase deficiency or fully responsive PAH deficiency. And so the BH4 medication called sapropterin would be used in treatment for this patient B, in addition to the phenylalanine restricted diet. So that concludes case 2. Moving on to case 3, this is the case of a patient that presents with pain in his right great toe and he is diagnosed with gout. Gout is a condition where uric acid precipitates in the joints and causes pain. There are 3 different ways that gout can present, either by decreased salvage of purines, overproduction of purines, or underexcretion of uric acid. In purine degradation, the base is excreted in the form of uric acid, and the uric acid is then excreted in the urine. Plasma urate levels in this individual are elevated, which would be characteristic of gout. And that doesn't help us determine between the 3 options because the plasma urate levels would be elevated in all these individuals. Low urinary acid levels are seen in this patient, which suggests a deficiency resulting in decreased excretion of uric acid. And hyperuricemias can be differentiated between whether or not the patient excretes normal or excessive amounts of urate. So option 1 is the overproduction of purines. An example of this would be von Gurk's disease. In this disorder, there is increased flux through the pentose phosphate pathway due to glucose 6-phosphatase deficiency, which results in glucose 6-phosphate becoming trapped in the cell. The substrate can be oxidized through glycolysis or through the pentose phosphate pathway, which will increase production of the activated sugar ribose 5-phosphate. This will then lead to an increase in purine bases. Total body urates will be elevated, and there will also be lactic acidosis. So testing a lactate level, glucose level, and white blood count would be other tests that could be performed in this individual. And there are multiple other problems associated with this disorder, so further testing such as neurological testing would also need to be performed. However, this is not likely to be the case of this individual due to his decreased level of urinary uric acid. Option 2 is the decreased salvage of purines. An example of this would be the Lesch-9 syndrome, and is a deficiency in the ability to salvage nucleotides. This is also characteristic of a dopamine deficiency. The HPRT1 gene is required for purine recycling, and a mutation in this gene leads to deficiency in hypoxanthine guanethine phosphoryl transferase. This would present as neurological symptoms as well. Option 3 is the decreased urinary excretion of uric acid. Another instance which can lead to this is excessive alcohol consumption. This leads to an increase in lactic acid production, and there is competing excretion for lactic acid and uric acid, which would lead to an underexcretion of uric acid. This could potentially be the case in this patient due to his decreased urinary uric acid levels. However, in order to differentiate in this patient whether option 2 or option 3 would be the cause of his symptoms, other information would need to be obtained, such as a medical and familial history, and whether or not he drinks alcohol and how much he drinks would also be helpful to know, as well as what other medications he is taking and what his dietary habits are. And in terms of the dietary change we do know about in this patient, which is an increase in drinking sodas, which would increase fructose consumption. As a result, this will increase purines. This happens by the following mechanism. Fructose is phosphorylated into fructose 1-phosphate. This is catalyzed by fructokinase. Next, aldolase B cleaves fructose 1-phosphate into DHAP and D-glyceraldehyde. When intake of fructose is increased, phosphorylation into fructose 1-phosphate is rapid, while the reaction with aldolase is slow. This leads to an accumulation of fructose 1-phosphate. Intracellular phosphate also decreases, which stimulates AMP deaminase that is responsible for catalyzing degradation of AMP to inosine monophosphate, which increases the rate of purine degradation and increase in uric acid. So this may have contributed to gout in this individual. And that concludes case 3.